Heavily requested topic is protein synthesis. Here's what you need to know in just over 60 seconds. By the way, this is triple only. For Edexcel, this is paper 1. For AQA, this is paper 2. Protein synthesis is the process of making proteins. Your DNA is what codes for proteins. Think of it as just a very long chain of code with sections called genes, which code for proteins, and also there's some non-coding sections too. That genetic code, your DNA, is stored in your nucleus, but proteins are made and synthesized in your ribosomes, which are outside of the nucleus. So this whole process of protein synthesis involves us first copying the code from the DNA in your nucleus and then sending it over to the ribosomes to be translated into a protein. Copying that code in your DNA is called transcription, and then translating it into a protein is called translation. Starting with transcription, so we're in your nucleus, the DNA is a double strand, so the first thing it needs to do so we can read the code is unzip. Then DNA polymerase will build a copy of that gene, the little section of the DNA coding for your protein, called an mRNA strand. That mRNA strand then leaves the nucleus and heads over to your ribosome. Now that up to there is transcription. Over at the ribosome, we're going to translate that code, the mRNA strand, into a protein. Within the cell, there are these things called tRNA molecules, and on those are amino acids, the things that we're going to use to build the protein. Each tRNA molecule has an anticodon on it, basically a triplet code, which will correspond to each triplet code that you find on the mRNA strand, which is currently sat at our ribosome. So complementary tRNA molecules are brought along to your ribosome, bringing with them a building block for the protein, an amino acid, one by one, amino acid after amino acid after amino acid, using those corresponding codes that the tRNA binds to on the mRNA strand. Now, when all of those building block amino acids are brought together by those tRNAs over here at the ribosome, they will then bind together to make that first strand that will form our protein. More complex things happen to that strand. It folds, more bonds are made. You don't really need to know that, but that is what then produces our final protein, something like an enzyme, for example. And that is translation. Speedy recap. DNA unzips in the nucleus. mRNA strand is produced that is complementary to the DNA. That process was transcription. Then over to the ribosome, the mRNA strand goes. Complementary tRNA molecules come along, bringing with them the building blocks for the protein, amino acids. Those are then bound together to produce our protein. That is translation. I really hope that helped. Make sure you save this video so you can watch it before your exam. Follow along for loads more videos to help you ace your science exams this year. And let me know if you have any specific requests in the comments, because I'm more than happy to make videos by request. Best of luck this year.